Today we're going to talk about Chapter 5. We're going to talk about cells. When someone has cystic fibrosis, one of the ways that they think that people get cystic fibrosis is they have a faulty CTFR gene. So that person would have cystic fibrosis if they had that faulty gene. So it's the failure of one protein to work right, which can cause this cystic fibrosis. And this, of course, this gene. So it's a defective CFTR gene that causes this, and the gene makes a protein that controls the movement of salt and water in and out of your body's cells. And so if you don't have that gene, that can't work properly. In people who have CF, the gene makes a protein that doesn't work well, and this causes thick, sticky mucus and very salty sweat. So we've discussed this before. The cell membrane is a phospholipid bilayer because it has two layers. And if you look at this picture, it shows good examples of phospholipid bilayer because it shows the blue heads of the phospholipid, which you know are hydrophilic, and then it shows the red, stringy, sperm-looking like tails in the middle, which are hydrophobic, which don't like water. And so you have a double layer, and of course you have proteins, which there's purplish-looking structures embedded within the bilayer. And the bilayer or plasma membrane is going to move substances in and out, and it's going to be selective. It's just not going to let anything come in and out. And osmosis and diffusion and vesicles are involved with the movement of substances in and out. And we already discussed the head and the tail and what their likes and dislikes were. The cell membrane, or the fluid mosaic model, will allow things to move in and out. And it's called the fluid mosaic model because there are little pieces meshed together. So a mosaic in art is when you take a bunch of pieces and put it together to make a picture. So that's why the cell membrane is also called the fluid mosaic model. And you have a diverse amount of carbohydrates and proteins attached to that, and you saw that on the last slide. And of course, archaeobacteria are going to have a more rigid membrane than eubacteria and eukaryotes. And on the right, I've just put a picture of a generalized bacterial cell, which you had on the last slide practical. If you look at this picture, you'll see the cell membrane or plasma membrane, and then you'll see the cytoplasm inside, and then you'll see the cell wall on the outside, and you'll see DNA, the spaghetti looking stuff on the inside, and then you'll see free-floating ribosomes, which are all those little green structures, and then you'll see the pili, which we've discussed, which is how bacteria can sexually reproduce and undergo conjugation. And then, of course, on the end, you have the longer flagella. The phospholipid hydrophilic head is going to love water, and it's going to point outward. And here's a picture showing you more detail. The polar head is phosphate containing, and then you have a fatty acid tail, which is non-polar tail, which is the part that doesn't like water. You have the phospholipid hydrophobic tail, which hates water and points inward. So you have the polar head again, phosphate containing, and fatty acid tail, non-polar tail. So the head loves water and the tail hates water. Hydrophilic versus hydrophobic. Okay, so you have different types of proteins, but you can sometimes have adhesion proteins. And an example is called a cadherin, and it's named for calcium-dependent adhesion. And so these play an important role in cell adhesion. And well, what does any of that mean? It basically means that your cells can't all be just completely rogue and completely separate from each other doing their own thing. They have to be attached to each other in some instances, in many instances, and they have to communicate with one another. And there's all these different types of junctions that occur inside cells. And so one thing that can occur is you have these cadherins, which help cells to adhere to one another so that they can work together. And in this picture that you see, there's an example of how adhesion proteins might be attached to one another. We've discussed enzymes a lot. Enzymes speed up reactions. They're catalysts. They have active sites. They bind with substrates. They're important in your body. Enzymes are important in bioluminescence, and in medical imaging and the use of dye. And if you look here, you'll see Pac-Man and pizza. And you also see a picture at the bottom about how enzymes can react or inhibit an enzymatic reaction. So there's things called recognition proteins, and those will do exactly what they say in the name. They're going to recognize molecules, they're going to find them, and they're going to bind to molecules. An example is peptidoglycan. You're also going to have things called receptor proteins, which are usually in the plasma membrane, and it's going to get signals from outside the cell, from the extracellular matrix, and it's going to use it for intercellular communication, like communication between cells from one cell to another. And this is important in synaptic transmission and hormone secretion. And we haven't talked about synaptic transmission, but just know that synapses have to do with neurons and have to do with your nervous system and getting those signals from one place to the next. And if you look at this picture, you'll see receptor protein. It looks greenish. And so that receptor protein is there within the cell membrane, and it's showing that it's getting chemical messages inside and outside the cell so that it can communicate between cells. So you have transporter carrier proteins, and during active transport you need ATP, such as in the calcium pump, and in passive you don't need ATP, and that's like facilitated diffusion. So if you look on the right, you're going to see carrier or transport proteins, and so you're going to see a protein, which is the purple structure. It's going to have a little channel down the middle, and it's going to grab the yellow substances from like outside the cell and pull them through the middle of the channel, and it's going to open up on the other side, and then there you go, you got the yellow molecules on the outside of the cell that have been moved to the inside of the cell. So when you have passive versus active transport, passive doesn't need any energy or ATP, and those examples are osmosis or diffusion or facilitated diffusion, and that goes with the concentration gradient, therefore it needs no energy. During active transport, you're going to need energy, which means you need ATP, so it's going to go against the concentration gradient. So you're going to go from low to high, and that example is a movement of calcium ions across a muscle cell, like the calcium pump. So in passive transport, you're going to go with the concentration gradient, just like if you ran out of gas at the top of a hill, you could drift your car down to the bottom with no gasoline. That's like passive transport. It goes from the high part of the hill to the low part of the hill. But for active transport, if you ran out of gas at the bottom of the hill, you wouldn't be able to drift up the hill. So that's from the low part of the hill to the high part of the hill. So that's active, and you would need gasoline or energy. So whenever you have passive transport, the examples are osmosis and diffusion. And if you look on the right, you'll see facilitated diffusion going through that carrier protein, like we discussed on one of the last slides. And if you look in the middle of the picture, or the middle of the slide, you'll see osmosis, which is the movement of water molecules from area of high concentration to low concentration until you get equilibrium reached. So in passive transport, you don't need energy or no ATP. Examples are osmosis and diffusion and facilitated diffusion. And facilitated diffusion is going to use a protein like the purple protein on the right. Diffusion is a type of passive transport where the molecules move from high to low until you get equilibrium. So it's going to be with or down the concentration gradient. And the rate's going to depend on size of molecules, temperature, concentration gradient, charge, and pressure. Osmosis is the movement of water from areas of high concentration to low concentration until you get equilibrium. And it's going to be with or down the concentration gradient, and it's not going to need any ATP, no energy required, because it's going to be going from high to low, so it's not going to need any energy. And if you look on the picture at the right, it's going to show you where you have some water moving from the left side to the right side across a membrane in the middle. Facilitated diffusion is going to use a carrier protein, and it's going to be passive transport, so it's not going to require energy, and it's going to carry those substances like we discussed before from the inside to the outside or the outside to the inside. So if you look at the blue proteins on the right, the protein is going to have a channel in the middle, and the molecules are going to go through that channel. So it's going to open like a mouth on one side, grab the molecules, move it through the channel, and move it to wherever it needs to go. And there's also gated passive transporter, which changes shapes. When you're discussing plasma membranes or cell membranes, they are selective, so they're selectively permeable, which means they can select and control the substances that go in and out, and they can control the amount, and it allows for maintenance of varying internal and external environments, so it can change what it needs to change by having that control. So it supplies nutrients, and it gets nutrients in, and it also removes waste, and through all of that, it also helps to maintain volume and pH.
So I have this concentration gradient analysis for you so that you can understand it. So when you have a difference in concentration between adjacent regions, you have a concentration gradient. So when I say that osmosis and diffusion and facilitated diffusion are where you move down the concentration gradient and there's no energy required, then I discussed the analogy of a car running out of gas. So when you go down the concentration gradient, like the truck on the right, you're going from the top of the hill down. So you can drift down that hill and it's not going to need any energy. So that's going to be an analogy of passive transport. And then if you look at the bottom, you have a car going up the hill. If it's going to run out of gas, then it's not going to be able to drift up the hill. So it's going to need energy. So at the bottom, that's showing you active transport, and the top is showing you passive transport. So when you have active transport occurring, you can have co-transporters, and that's the usage of protein to move two substances at the same time across the membrane. And an example is the sodium-potassium pump, which is the movement of sodium out of the cell and potassium into the cell. So if you look at the top, you're going to see what's called a symporter in the middle, and that's going to be the A and the B moving across the membrane at the same time in that one direction there. And then you can have an antiporter, which would be one moving one way and one moving the other way. And then you have a uniporter, which is one substance moving across in one direction. Vesicles we've discussed in class with the animal model that you looked at. Vesicles are substances that aid in the movement of products into and out of the cell membrane. So the vesicle is going to move toward the surface of the plasma membrane, and it's going to cause an opening in the plasma membrane and release those substances into or out of the cell. So it's that indented part of the circle that you saw in the lab on that purple model. So whenever you have endo and exocytosis, endo is going to be something going into the cell and exo is going to be something going out of the cell. So endocytosis is going to use a vesicle, it's going to enclose a material, and it's going to cap in at the cell surface at that cell membrane we've talked about before. And it's going to bring products into the cell, either liquid or solid. And receptor mediated is when specific molecules bind to surface receptors enclosed in a vesicle and they bring it into the cell. If you look at the picture at the top, you'll see a membrane and you'll see a food particle being brought into the cell. That's a little diagram that shows that. And then you'll also see a vacuole, which will have reversing it and it'll show exocytosis. So the top picture shows endocytosis and the second picture shows exocytosis. And then down at the bottom, you'll see exocytosis. So you'll see a vesicle and you'll see it moving toward the cell membrane and then releasing its products to the extracellular fluid. So exocytosis is fusion of a vesicle. It occurs in the cell membrane and it releases those contents out, either liquid or solid. So you have three pathways for endocytosis. You have bulk phase endocytosis, receptor-mediated endocytosis, and phagocytosis. So if you look at the picture at the bottom, you have endocytosis. And if you're taking in food particles, that's going to be phagocytosis, and it shows it on the first picture on the left. And if you have pinocytosis, it's going to be liquid. So phago sounds like food, and then pino, I always tell students, it's like little pin drops of pop on the top of a pop can. So pino is liquid and phago is food. And then you have receptor-mediated endocytosis. And if you look at the diagram, it'll explain that, where you have these coated proteins and a coated vesicle, and then they are going to see a receptor there, and it's going to recognize it and bring whatever it needs to into the cell. So in a cell, you have tonicity, which is the type of solution that you put a cell in. So hypertonic, isotonic, and hypotonic are the different types of tonicity. So when we did the gummy bear lab or the gummy worm lab, we were experimenting with different types of tonicity. So hypertonic is when substances go out of the cell, and that shows at the bottom with the green arrows going out of the blood cells. So if you have a hypertonic solution, the cells are going to get smaller, and eventually if they get too small, they can die, and that's called plasmolysis. Isotonic, the cell is going to stay the same size, so the same amount of liquid is going to go into and out of the cell, like with the arrows at the bottom. And if you have organs that are going to be transplanted, you're going to put them on ice, which is how you can remember that, and iso means same. On the right, you have hypotonic. Hypotonic is like blowing up a balloon. If too much air goes in the balloon, it's going to burst. So in a hypotonic solution, if too much liquid goes in, then the cell can burst, and that is called crenation. So plants have turgor pressure or hydrostatic pressure. So when you water a plant and it's not wilted, then the vacuole has turgor pressure, so it has plenty of water. But if you have a plant that gets wilted, then there's not enough water in the vacuole and they lose turgor pressure and they wilt. If you have a plant that is so wilted that you can't water it enough to get it to become unwilted, then it means it is permanently wilted and it's not going to come back, which means that it's lost turgor pressure. Way too much for you to get it back. So the last slide has to do with osmotic pressure, which basically just means water pressure. So you have a solution moving around, and as you have water flow from high to low or low to high, you're going to have a change in pressure.